person, I think the art and scientific endeavor are very similar in that they're both technical and they're both creative, and I think very visually, as you'll probably see from my talk. But since I was a little kid, I've loved modern art like this, which is hanging in the National Gallery where I'm from. And I was always really inspired by modern art in its ability to kind of sample what I like to think of as the pattern space. And so I was inspired by that in my own career in thinking about how nature is able to actually sample the pattern space. So this is a really beautiful artist rendering of the cytoplasm inside of a cell, which is actually probably not too far off of what it looks like, which looks very similar to this, I think. And so I think about this problem. My answer is not working. It's not working. Okay.
They have it on the board.
Oh, that's okay. Go back and forth. I think about this problem at the cellular level, and so probably you've all taken high school biology or some form of biology, and you may have learned about cells, and you may have thought and learned about cells as these bags of water in us. But cells are actually really beautiful and amazingly patterned. So here's two examples of single-celled organisms. This is a kind of marine algae. This is a single-celled protozoan that have these cool hair-like projections off of it. And so this is, hopefully you're amazed by that. Wow, look how beautiful these cells are. I am. And this is not just true of single-celled organisms, but it's also true in our bodies. So we have cells in our bodies that are similarly elaborately patterned. So, for example, this is a cell from our lung bronchiole, and this is one of our neurons, which we're going to hear more about in the next talk. So cells can achieve these amazing diversity of form, which is really intimately linked to their ability to do different functions in our body, basically. And so, it's not just that this patterning is fundamental process, but it's also linked to disease. So for example, this is a cell in our cochlea, in our inner ear, and this is a single cell, and all of these little structures are little things called stereocilia sticking off the cell. And the actual patterning of these structures is really how we can actually hear different frequencies in all the sounds that we hear every day. And so we know that there are diseases and also external stimuli that actually can perturb this patterning that will lead to effects on our hearing. This is just one example of how these defects in cellular patternings can lead to disease, basically. And so I'm really interested in this question of the cell and developmental biology of how cells achieve patterns. And the kind of main takeaways of my talk today, let's put them up front so you know where we're going. One is, as we've already talked about, cells are incredibly diverse in their form, and this is, again, intimately linked to their function. And we're going to talk about the structure called the cytoskeleton, which is awesome, and is a fundamental aspect of the cellular diversity that we see. And two really important things to me that I like to really put out there is that one, I'm a basic science researcher, so I study fundamental problems. And this kind of basic science research is really what leads to major breakthroughs that actually directly impact human health. And these discoveries usually involve studies in model organisms, as we'll talk about. And importantly, like very fundamental aspects of biology, despite what you may have learned or seen in textbooks, are really unknown, which is kind of amazing, is that really the fabric of what we are is really unknown. And we can't really understand disease until we really understand how normal physiology works. And so this is what inspires me every day to be able to go into the lab and do what I do. Okay, so then back to this question of how do cells achieve this diversity that we see here? And so underlying this structural diversity is something called, is kind of a molecular skeleton in the cell called the cytoskeleton. And so these are two examples of cytoskeletal elements. In red is something called actin, which we're not going to talk about today, and in green are things called microtubules. So in all of our cells right now, we have these structures. And again, the cytoskeleton is really important for patterning the cell. And so what is a microtubule? So if we zoom in on one of these long polymers, we can see that it looks like this. It's a really nice long polymer. It's composed of these little subunits, which are represented by these little green circles. And these polymers are actually really dynamic. So under that is a movie of individual microtubules kind of growing and shrinking in a test tube. So this is also happening in our bodies right now. Microtubules are growing and shrinking. There's also special proteins that help to stabilize them so they don't do this, okay? And microtubules have many functions in cells. So going back to that cell that we just talked about, this lung bronchiole cell, all of these structures that stick off the surface of these cells are made of microtubules and allow for, wow, the resolution is really bad on that video, but these structures beat and allow for us to clear things from our airway, for example. This is actually the surface of a protozoan, a paramecium, probably you may have looked at it as elementary school kids, but these structures are the exact same as these structures. Like we have the exact same thing sticking off of our cells that weird cells in ponds are swimming around with right now. And so these structures in both cases are made of microtubules. Microtubules can also generate force and movement in other ways. So this is a cell that's dividing. So in black here are the chromosomes in the cell, in red are the microtubules, and microtubules are literally reaching into the chromosomes to divide the chromosomes equally so that each of our cells gets a full complement of DNA, which again is imperative for that cell to go on to survive. And then also, one of my favorite things about microtubules is that they are literally, right now inside your cells you have microtubules that are these molecular highways on which proteins called molecular motors, which this one's called kinesin, are walking right now. In all of our cells, they're walking and carrying all these crazy cargoes from one part of the cell to the next part of the cell. So microtubules structure, dynamics, and then also the cellular highways that are allowing for the transport of information in cells right now. Okay, so in order for microtubules to do all these really cool things, they need to be actually spatially organized, and that spatial organization is again what allows for this kind of cellular diversity that we see. So here's some examples from our body. We have different kinds of epithelial cells, which are the cells that line our organs and provide barrier function, neurons, and skeletal muscle cells. And if we look at the microtubules in these cells, hopefully you can appreciate that microtubules are organized in very different ways depending on the cell type that we're looking at. And this organization is, again, critical for their function. We'll hear more about neurons in the next talk, but microtubules going down the length of axons and dendrites oriented in a specific way allows for information to travel in very specific ways. Okay, so how do microtubules become organized in cells? This is one of the only jargony things that you'll have to remember. There are these sites in cells called MTOCs, or microtubule organizing centers. The best studied MTOC is something called the centrosome, so this is now a one-cell embryo. You have the spindle that's being built that's going to, again, divide the DNA up. And at the center of the poles of that spindle is the structure called the centrosome. And that's organizing microtubules into these radial arrays. And it's doing so because there's physical stuff, this blue stuff, that actually allows microtubules to grow into these radial arrays. Okay? And so people have studied centrosomes for 100 years. They're super cool. I've studied them for a long time. But they're not the only game in town. Because in different kinds of cell types, both in the same organism and in different organisms, microtubules are organized in very different ways. And so this radial array stuff doesn't really work. So when you need to build, for example, parallel arrays off a specific site in epithelial cells, you have MTOC function imparted to a different part of the cell. And we know much less about how this works. And this is one of the main focuses of my lab, is to try to understand how this MTOC function is imparted in cells. And what we know is that there's a shift in MTOC function that happens in development and also normal physiology. So this is now a one-cell embryo. Again, it's dividing. So in most embryos, including us, first we were started as one cell, and then the cell is going to divide. And usually those divisions are reductional, meaning that every cell is smaller and smaller and smaller. And that's the first phase of development, until you get to a stage where cells begin to specialize or differentiate. And that's an example of this embryo is an example of that. So it's starting to build different kinds of cells within it. And these cells are much smaller than this one giant cell. And so each one of these cells is going to be an epithelial cell that has microtubules organized off these apical surfaces here. And so hopefully you can see that there's this big shift from this kind of centrosome-dominated array to this kind of non-centrosomal, or in this case, an apical MTOC. Okay, and so this is, again, a fundamental process that's going on in development. Right now in our body, as cells renew themselves in our intestines and liver and all these things, the cells are having to reorganize their microtubules. And so this is just a part of normal physiology, but it's also something that can go arise in disease. This is another reason we are interested in this topic. And so it's been known for a long time that if we look at epithelial tumors, so most cancers are derived from epithelial cells. And if we take a section through normal healthy tissue, and we look at the microtubules, we can see a few microtubules associated with the centrosome in this breast tissue. But if we look at this epithelial tumor, we can see that microtubules have gone crazy. The centrosomes are overactive, there's crazy numbers of microtubules. And so this has been noticed for a long time, this correlation. And more recently, people have shown that this overactive MTOC function of the centrosome actually leads to the cellular behavior that allows cancer cells to become migratory. So oftentimes, you have epithelial cells that are contained, and the cancerous scission involves a transformation of the cell to become more migratory, so it can move through your body and metastasize. And so that movement behavior is actually associated with this overactive MTOC function. So we think by trying to understand this fundamental process, we can understand new targets for example, cancer therapies. Okay, so then one of the big questions that we're studying in my lab is how do microtubules grow and become organized in different cell types? And this, again, is a very fundamental question which we think has direct human health applications. And so we know something about this topic. So if we look at the Bible of cell biology, which is called Molecular Biology of the Cell, this is the textbook I used to teach cell biology here to our undergrads at Stanford. And it's a lovely textbook, and like most textbooks, the things are not always right. And so if we look in how microtubules grow in this textbook, it's going to tell us there's a structure called gamma Turk. This is a essential complex of proteins from yeast to humans. It was actually discovered in a fungus, but it's conserved. It's essential. You get rid of it in organisms, they all die. So it's very important. And what it is this little complex of proteins that form a ring, the gamma-tubulin ring complex, that allow microtubules to grow from one end to the next. Okay, so they template the assembly of a new microtubule. And so we know the gamma Turk is important. Again, we get rid of it, things die. And that's true. This is a one-cell embryo with this beautiful spindle, and white are the microtubules again. We get rid of gamma Turk, the embryo, the spindle is crippled. There are way less microtubules. Again, the embryo dies. But there are still microtubules here. And you actually have to get rid of gamma Turk and another protein called Aurora A to really get rid of all the microtubules. And that's all. The protein names don't actually matter, but just the point is there are things that are acting in addition to gamma Turk. Okay? So we started out with this problem, this question of how microtubules grow in different cells, and we understand that the centrosome, it involves this protein gamma Turk. That's what the textbooks say, great. So we have this hypothesis that maybe it's these same proteins that build microtubules in the centrosome that are required to build them at other sites. Okay, so how do we actually study this problem or test this hypothesis? Well, so for us, and people that think and care a lot about the kind of native patterning of a cell, we can't actually study this problem in a dish because we know from numerous experiments that cells grown in a dish behave very differently than normal cells, partly because many of the cells grown in a dish are derived from cancers. So if you care about something like how a cell is patterned, you have to really study it in its native context. And so we wanted to observe this process, so the process of how cells differentiate in its native context. And then to learn how it works, we need to be able to break that process and then repeat, basically. So you break it, observe it, break it, observe it, until you try to understand how something works, right? And so how we actually do this is, so to observe, things are usually small, we use microscopes, but we need a model organism to do this because we can't, again, study this in a dish and we can't study this in our cells because that would be unethical and also hard. So in order to break the process, we need genetic tools to be able to literally manipulate gene function. And again, this points us to a model organism. And then we need, of course, patience because science is actually a laborious process, unlike what shows with CSI and things like that show you. Yes, it's a very snail pace. Okay. All right, so what is our favorite model organism? We love this little worm called C. elegans. Has anyone heard of C. elegans? Yes, that's great. Okay, so C. elegans is a wonderful model organism. It's a small, this is a 1 millimeter long worm. It's a nematode roundworm. And it has great features. Okay, the worms and their embryos are transparent. The genetics are awesome. So worms are hermaphrodites, so they make egg and sperm. So without doing anything, a worm will make 300 progeny. You don't have to do anything. They can survive desiccation. You can freeze them. They're wonderful for grad students who just leave their plays out to die. So, they have fast reproducible development. Okay, and so that's great because we can use the transparency of them to do microscopy. And I'll show you more about that. We can use the facile genetics to do genetics. And the fact that they grow fast means we don't need a lot of patience because the generation time is 3 days. So that's awesome. So to make a mouse, it takes 3 months. To make a worm, it takes 3 days. So it's very wonderful, but it changes the kind of scale of problem that you study, basically. Okay, and not to brag, but there are 6 Nobel Prizes for people working in C. elegans, which is great because again, it just highlights how, again, fundamental processes are really important for human health. So if we actually look at the Nobel Prizes awarded for medicine and physiology, 160 of 216 actually use non-human organisms in the research. So hopefully this just underscores the fact of how important it is to kind of support basic research and also basic research in a model organism. Okay, so back to these questions. How do we observe something in our little worm? So we can use something called green fluorescent protein, GFP. Who's heard of GFP? Awesome, yes. So GFP is amazing. This didn't happen in my scientific lifetime, but it changed the world of science for sure. So someone who was very interested, who's seen bioluminescence? Anybody seen? Yes. Bioluminescence is awesome. You go out in the water, there's these things floating around, they're fluorescent, you can see them under the moon, it's amazing. So jellyfish are bioluminescent. I was just reading, actually, the sharks are too. That's what they just figured out. And so someone was interested, why are jellyfish bioluminescent? And so a researcher figured out that there's a protein in jellyfish that was called green fluorescent proteins that literally makes the jellyfish fluorescent. So then someone else thought, aha, I'll take this GFP protein and I will put it in another organism, like a worm, and I will make that worm fluorescent. That's exciting. And then we can do things like make all different kinds of fluorescent proteins, and now we can make the worm all different colors. We can make whatever organism we want all different colors. And that also got a Nobel Prize, because it's awesome and it transforms science. And then, so we have to ask ourselves, so we have this GFP tool, how do we get it into our organism? That's an important question. We used to have to do all kinds of witchcraft to get the GFP in, but now there's CRISPR. So who's heard of CRISPR? Probably many more people have heard of CRISPR, right? So CRISPR has changed science. This is, in my scientific career, this is the most exciting discovery that's happened. And again, came from interest in basic science, because this came from someone studying bacteria and the way bacteria can defend themselves against the integration of foreign genetic elements. And so using that technology, CRISPR and Cas9 is a protein that cuts specific DNA sequences that are programmed by their CRISPR sequences. We can use the same technology in our organism to then cut the DNA and insert whatever we want, or cut the DNA and make whatever mutation we want. So that's exciting, because now we can just take the DNA that encodes GFP, we can put it in our genome, and we can look at how the natural proteins in that organism look and how they behave. And we do this, we literally just inject the worm, which is harder than it sounds, or maybe easier than it sounds, I don't know. And we can make everything green. So this is what it looks like. This is a worm embryo. And so what you're seeing here, this is the microtubule that I showed you, remember it has different ends. So this is a protein that binds specifically to the growing end of a microtubule. And so that's exciting. So this is early in the embryo where you have these centrosomes here that are at the spindle. And so hopefully they look like beautiful comets emanating from this centrosome. And then as the embryo develops and the cells start to differentiate or specialize, we can see now these other surfaces growing microtubules off of them. So this is the intestine. This is the surface of the embryo or the skin, and microtubules are growing along the surface. Okay, so we can just literally, in this worm, watch microtubules growing, which is awesome when you're trying to observe this process, because we can just observe the natural state of microtubules and what they're doing, which is really exciting.
What's the time scale on these videos?
This is a little bit sped up. I think this is like 5 times not normal timing, basically. But good question. Yes.
Can you turn them off and on, or they're all on?
So we could do that with genetic perturbations. So this is what we've done here is we've inserted a protein that's fluorescent, and we've fused it to another protein. And so this is just what the natural protein is doing. Oh, you're saying can you turn off the GFP off and on? Yes.
You attach it to the ends of the microtubules,
Yeah.
right? So all the microtubules
Yes.
are glowing.
Well, so all the growing ones. So this is a protein that labels specifically growing ones, yes. But if you can turn it off and on, we can use tricks to then say wherever the GFP is, let's stop the microtubule from growing. Or we can bleach the GFP and actually make the fluorescence go away, and we can learn things from that too. So we can also do fun things. I'm going to run over my time, but it's fun. We can also do there's different kind of fluorescent proteins that you can actually change their color. So they start out green, and you can shine light on them, and they will become red. And that's really cool, because if we want to mark individual comets and say what happens to them, we can mark one, change it to red, and then over the lifetime of that molecule, we can say what did the red one do in this field of green things, basically. So there's a lot of really fun tricks that exist these days. Okay, so we have a lot of tools. C-Elegans is awesome. Hopefully you got that message. And so what are we going to study? So one thing we study, one tissue we study is this embryonic intestine here. So this is where all these comets were, and the comets were emanating from the surface. These are the apical surfaces of all these cells. There's microtubules are growing. And lo and behold, the gamma Turk, that really important protein, is localizing to that surface. So that's really important, because it seems like if it's there, maybe it's playing a role. Isn't that a good idea? And this is what, if we blow up this region, this is what it looks like. So microtubules are here, they're growing off the surface. This is the nucleus, this is the centrosome here. Centrosomes are inactive, but microtubules are instead growing from these apical surfaces. Okay, so here's the problem with when you work in an organism, right? So if we get rid of gamma Turk, it's essential for mitosis, so everything dies. So we can't, the embryo literally doesn't make differentiated cells. So we can't ask the role of gamma Turk in differentiated cells if the thing is not alive. So we needed a way to be able to specifically deplete gamma Turk from a tissue of interest. So that's what we did. We used CRISPR again. We can insert something called a degron. All a degron is another piece of sequence that when a degrading protein sees it, it tags that protein it's attached to for destruction. Okay, so we inserted a Dagron and GFP. This is what it looks like. Again, the gamma Turk is on this apical surface. We can blow up that intestine here. And now, there's no degrading protein expressed here. Now we can express the degrading protein specifically in the gut, and we can remove gamma Turk specifically from the gut, which is awesome. Because now the embryo is alive, it got to where it needed to get, and then we depleted the protein of interest. So that was, for us, this is a huge boon because now we can do all these kind of experiments. And so we were incredibly surprised to see this result, which is that the embryo, these cells don't care if they have gamma Turk. So this is a control embryo. You have all these microtubules growing off these apical surfaces, and this is an embryo where gamma Turk is depleted from the gut. And so hopefully you can see that there's really no difference here. And there's tons of microtubules despite the fact that the only complex we know that makes microtubules is now gone. So this was like exciting, and also, it's the first, really? This is one of the first things I did when I started my lab here. This is an easy experiment, let's do this. And then the surprise is that things don't work like we think we do. Okay, in the last, I think I have 1 minute left or something? Yeah, so in the last minute, I'll just tell you kind of what we're doing moving forward, which is new technologies, which is, okay, so we still don't know what is required to build and grow organized microtubules in these differentiated cells. We know that gamma Turk is not the whole story, or much of the story. So the question is, what is there? So we know gamma Turk is there. And we know that there's this other protein called Patronin there. The names, again, don't matter, although this was named after Harry Potter for the Patronus charm because it protects microtubules. That's an aside. Anyway, so we think what's happening is, so these proteins are localized there, so they probably associate with some of the microtubules, but in fact, there's probably a whole bunch of other proteins there that we don't know what they are that are actually building the majority of the microtubules. So how are we going to find those? So to do this, we collaborated with this awesome woman on campus, Alice Ting, who has a lab here as well. And she's a biotechnologist, and she evolved these enzymes called TurboID that what they can do is you can, like GFP, you can just attach it to your protein of interest. And what it does is it takes this small molecule, this vitamin called biotin, which maybe you've seen in like your shampoo and stuff. And so what it'll do is it will fuse biotin to all the proteins that are close to what's called your bait protein. So that's cool. Because now we don't need to know anything. We can just take Patronin, which we know Localized is there. We can stick on this enzyme, and it's going to stick biotin onto everybody that's around. And then we can just pull on the biotin and pull the things that have biotin down, and we can sequence the proteins that came out. And so doing this, we found a ton of proteins, which is exciting. All of them all the ones we cared about are conserved with humans, which is exciting. And more exciting is that we can then repeat that process where now we deplete them. We do genetics, get rid of them, and now we can start to perturb microtubule organization. So that's very exciting. This is our entry point, is to try to understand how this process works. And so this is just the beginning of our journey. I have more to say about this if you care, but we have lots of information about this. But just again to highlight, we started with this really basic question of how do you build the cytoskeleton in the cell? We used a model organism. We did observation and to try to find these fundamental mechanisms, and with the ultimate goal of really trying to understand disease. And so those are the main points I want to leave you with. And so I would just like to thank my lab. This is many of them here. It's a wonderful, being a scientist is really wonderful, and the people that you train are inspiring. And these are all the people in my lab now, and some of the former people that contributed to this project, and our collaborators and funding. And so I thank you for your attention, and I'll take any questions.